Okay, so we'll begin. And we had started this discussing RNA sequencing in the last class. We will finish it, and hopefully we'll be able to finish. It's not. Um, it's probably not the most technically challenging part of the course, but this area in itself, there are a lot of innovations again that have happened, which we will highlight as we go along. Um, before we begin, I think there was one question about from I think Devansh, not here anymore. Oh, he's here. Yeah. About this Ultima sequencing, right? So I think I'd said something wrong, which was I've seen paired end versions of it. So Ultima, you're right. You will use it as a long read sequencing throughput. So it's only one end that you end up sequencing. So it's actually correct that it only sequences one end. Because a paired end is not required in the sense that you're already getting something which is 400 nucleotides long, and also the design actually does not allow for a paired end sequencing. So that part is correct, right? Because the way the chemistry works is all of it is flowing in one direction. There is no bridge amplification kind of thing happening, so there is no switchover that happens. Does that answer your question? Sorry, I think I said it allows paired end, but it doesn't. What? The question that you had was, does Ultima sequencing, Ultima seq, which is a $200 genome, does it allow for a paired end? Is it only single end or is it paired end by design? It's only single end by design. Okay, any other questions from anything we discussed last time? I think you've already started doing your assignment. So should be, I think I got one question that it should be, the BWA should be run on each word in the text, but you'll see that you reuse the meaning if you run, right? So life, death, whatever. So the reason we are running VWT on this text is because we are running it as a compressor, right? At this point, all we care about is how we can compress this data. So if you can run it on each word separately, but there's nothing that's going to be very useful, right? Because these are all small letter words. So even if I run BWT on AND, what will it give me, right? Like it will give me the same thing actually, or ADN, whatever, or whatever is the corresponding sorted version or the cyclic version. So that we don't care about. What we care about is if I put this all together, what does the final transform look like? And there's a specific pattern that will emerge, which you have seen also. It's not very surprising, which is the reason this works, which is the reason compression works. So that's why I ask you to run it on the full text and not on individual words. Okay. And yeah, how will you handle spaces? Yeah, so you can give it a particular character that you want. You can also keep it as space. It just doesn't matter in this particular context. Just have a special thing that you would like to assign. Any other questions? It could also be, for example, you could replace this with underscores and everything is taken care of automatically. Any other questions? Okay, so we'll begin with just going into a bit more details. So just a review that only 4% of our transcriptome, which is your RNA, codes for proteins. Everything else is functional. It makes the rest of the proteins function in a particular way. And this broadly is what is taught as central dogma, but this is not central dogma. It just says that genome gets transcribed into transcriptome. Transcriptome gets mapped into proteome. And the reason we are interested in sequencing this particular part is because this part is shared. It's the same across all the cells, whereas we know the protein is not, right? So my hair has a different architecture because it has special kind of proteins as compared to my liver or my kidney or my skin, but they have exactly the same DNA sequences. So an ideal world, we would be sequencing proteins, right? But we haven't arrived there yet. We cannot yet sequence proteins. We are getting very, very close. And maybe I think in the next 2 years, whatever I'm teaching will become defunct. And then I'll have to reinvent a new course. but we are not there yet. We are not being able to sequence proteins at the scale that we would want. The broad field of proteomics tries to do this by essentially working with a very small set of samples. That broad field is called proteomics. This course is not proteomics. That's why I discourage also all projects related to proteomics, because it's a different science and altogether. The focus is at the molecular level, can you detect what is happening, right? So proteomics right now as it exists is not actually sequencing the proteins. It's just detecting the amount of proteins. You don't know what is the protein being made up of in the way the proteomics is done right now. So the next best thing that we can do is, if we cannot sequence the proteins, is we go one level up and see that what makes the identity of these proteins are these copies of RNA. And that's where broadly is called the field of transcriptomics. So study of genome or DNA is genomics, study of transcriptome is transcriptomics, and of course proteome is proteomics. However, the omics that you compare across all these three domains is not comparable, because right now as I speak, most technologies do not sequence the proteins, whereas what we are focusing is we are able to sequence the genome, we are able to sequence the transcriptome, and when we sequence it, we are also able to tell you how many copies of each gene you have, right? So protein abundance, right? So proteomics, what is the broad theme in proteomics? I get cancer, for example, I will take my tissue, I will compare it with let's say I get cancer in my liver, and I take one part of the liver which is cancerous, I take a matched normal which does not have cancer, I will do proteomics. It will tell me that you have these 10,000 proteins which are present at let's say this is your protein 1, this is your protein 2, this is your protein 3. It will tell me that this is C and N. It will tell me that this has 10 copies, this is 5, this is 10, this is also 10, this is 15, and this is also 15. And you will say that, okay, you have cancer, that's probably because, or you got this particular gene or this particular protein to be differentially expressed. What I would never know is how did it come about to be? What led to that change? Or I would also not learn about, is it causal in some sense? Did I get this abundance change and that's why I got a cancer, or that I got a cancer and then that's why I saw this differential abundance, right? So genomics and transcriptomics are slightly more granular. They will try to be slightly more, they are also associative. We are trying to do association studies here in some sense, but there is slightly more context there, because I know that when I sequence this thing, what did the sequence makeup look like in the normal cells, and did that sequence makeup looks slightly different? Because if I see a change happening, then I can put, I still don't know whether it is associative or it is causative, but if you see a change that is happening in DNA, you can go one level up and see that maybe this is the reason it's causing that particular change, right? So that's why there's more granularity in these two fields. Okay, so we have to do some biology, which we started last time. So you have DNA, which is packed inside the nucleus. DNA will get transcribed through this process of transcription. There's an RNA polymerase. It's an enzyme which will read the DNA and literally copy, make it a transcript, make it an mRNA, make it an RNA, excuse me. Now what happens essentially is, this will become clear in the next few slides, is there is a process of splicing involved. So when the RNA polymerase makes the transcript, it's in a premature form, like so inside here, it's literally copying everything. But then at one point, there is a process which happens because of splicing machinery. It will go here and it will start to remove things which it thinks is not important, because not entire DNA codes for protein. There are certain regions within the DNA which are called exons, which make your protein. We'll see what are these exons in just a way. Once you have your processed messenger mRNA, this is exported out. And this can then be taken to ERs, which is your endoplasmic reticulum, for starting and doing all sorts of routine machinery. But for now, we'll stay very, very close to the nucleus. So this is what we talked about last time also. This is what a gene looks like. It has a special start signal, which is called the initiation codon or the start codon, and a termination codon. Codon is nothing but 3 nucleotides. We saw that how 20 amino acids are made up of these 64 combinations possible. That is called the codon table. One codon means 3 nucleotides. All of them are in frame. So the machinery in this case, for example, how do I make protein is I will have a ribosome come here. It will move 3 nucleotides at a time, keep going, keep going, keep going until it encounters a stop signal at which your protein synthesis is complete. So only the protein coding part is not the gene. The gene is basically anything which is getting transcribed broadly. There is a lot of debate about what constitutes a gene. But very, very broadly for this class, what constitutes a gene is the boundary at which you can detect a transcript. Okay. What is a transcript? So a transcript is nothing but the mRNA or the RNA copy version of this DNA. Sequences are the same ACTG, except that in RNA there is no T. A T is replaced with uracil or U. For all practical purposes in this class, that change does not matter. So I will use interchangeably a T and a U, but there are some special reasons where it matters, but for this class it doesn't really matter. What you see here, right? So this mRNA copy is what the RNA polymerase will make. So DNA gets converted to RNA. Why is it called mRNA? We will just see in just a bit. But overall, there is this particular part which is the untranslated part, or the UTR. This, even when the ribosome sees it, there is actually mRNA here, right? If there is no gene corresponding to here, this part will not even make mRNA or RNA. There will be no transcript that originates from this part. From this part, there is a RNA originating, but it is not getting translated into protein. Okay, those are called untranslated regions, or UTRs for short, and they're both in 5' and 3'. Yes. What is it? Yeah, sure. So the green area is overall which is going to make the protein. So the protein production begins here and ends here. Okay. This is the boundaries at which mRNA is being expressed. So this, this is your DNA. Only part of the DNA gets converted to RNA, and only a part of this RNA will actually end up making the protein. It is part of the DNA, yes, but you see mRNA copies come up from here. And when I show you the next slide, it will become clear what it is. Any other questions? Okay, right. So this is what actual gene overall looks like. So a gene broadly is determined based on presence of these exons. These exons are special sort of sequences which make proteins. And whatever you're seeing in between two exons is what's called an intron, right? So on basically comes from that codon, similar word. X, because this is external and this is internal in some sense, because this can exist towards the out. I don't know if that is the actual story of why they're called exons and introns, but I should figure out. You have multiple exons making up one particular gene, right? So there's gene exon 1, 2, 3, and 4. Overall, when the transcription begins, it can begin from anywhere, right? Like it can begin from here. Why is this exon important? Because the first exon usually will sit next to this region, which is called the untranslated region, which I showed you in the previous slide, which will not make protein, right? So you can have just before this region, you will have certain part of it, which is, even though it's making RNA, right? So this is alternative transcription start site. Your RNA production can begin here. But this part, even if it gets transcribed, which means it's an RNA, which will not become part of the protein. Protein only come from these exonic portions of your region. it skips the mid parts, right? So the introns are what's called, they get spliced out in the process. So you can see now this has become a combinatorial problem. How does the splicing happen? So overall, you have 4 exons in this case. At each point, I can choose which ones to retain or how to retain them, or what to make out of it, right? So the first example is, well, I splice this out, splice this out, splice this out. Intron, so get removed. You make this 4 exons join to each other, and you will synthesize this as one protein, right? So you'll get one particular kind of protein from here. I could have also spliced out, well, this is in the reverse direction, so that is very similar. Overall, at each step, I will show you what the splicing overall looks like. But each point, you have a choice to make based on which particular isoform. These individual ones are called what are called isoforms, right? So there are multiple isoforms which are possible from the same gene. A gene is made up of exons and introns. Introns get spliced out when you form the messenger RNA. Sometimes it's also called, colloquially called the mature RNA. Mature RNA is any RNA which has introns. So the first time you make an RNA copy, the introns will be there. But then the splicing machinery will come and will throw away the introns and will start joining the exons. And that's what you get overall towards the end. And the reason this is, it will become clear why it looks like this in just a bit. But overall, from the same possibility, you can make multiple possible mRNAs overall. That's what this plot is trying to show. That one single gene can give rise to many mRNAs that are possible. It has no. The start and the end are fixed. That's a good question. Exons are not characterized by the start codon and the end codon. Once I join them, this should be start and this should be the end. That's how the joining happens. How do we understand? Oh, good question. We'll see this in just a bit. How does it understand what happens, right? It's extremely smart, but it's a pattern matching algorithm that the splicing machinery runs to figure out whether it's an exon or it's an intron. The short answer to this, I will come to you. The short answer to this is introns are generally very A rich. They have a lot of A's, and it's able to figure out what are the splicing sites at which needs to be thrown out. So that's how it recognizes. There was a question that side. Isoforms, yeah. Yeah. So whenever you make, yeah, fair enough, right? So you have a choice to retain an exon or retain the intron, right? So what is the machinery doing? So you see 2 introns here, right? So it will become clear once I show you the next slide, but very briefly, you can think about that this machinery is looking for all these junctions. So if your machinery part 1 sits here and part 2 sits here, it's going to splice this entire thing out. And that's what happened in this case. That's how 2 and 4 got joined overall, right? And this could have happened to anything. I could have also spliced out exon 2 because one end of the machinery would have sat here and the other one would have sat here, and this would have gotten spliced out. So depends. Well, in this case, you cannot. Usually your exon 1 will remain, which is your first exon. Last exon often can get spliced out, but usually you can think that because that has to have that special stop signal overall, it will usually be retained. But there are always, in biology, there are always examples where it gets spliced out. So that can happen, right? Because it's possible that once that gets removed, this particular exon also has an in-frame stop signal. So we can try to find out if there are instances like that, but usually 1 and 4 will get retained. Everything else sort of happens combinatorially. Yeah. That's because of the length of the untranslated region. So the length of the untranslated region is, that blue part is how much of it is not being translated into proteins. That is actually also a very context specific thing. That's why in this case, it matters because it helps you stabilize the RNA. RNA is one of the most unstable species in your cell. So while this looks like nothing is happening there, there's actually a lot of magic happening there. So these are actually not linear, right? That's one of the number one mistake that a lot of us do. That while I'm showing you this in the linear world, it's actually a lot of it is happening in 3D. So in fact, what is happening in some cases is this end and the other end is talking to each other. And this becomes a site for other proteins to come and sit, which are called RNA binding proteins. So how long your UTR is, particularly towards the 3' end, helps you actually stabilize your RNA. So it does have a meaning. For this particular cartoonish picture, it doesn't matter. But if there's time in the class, we'll talk about one technology which tries to sort of look at that particular thing, and where you'll see that UTRs are actually very, very important. No, isoforms are mostly, isoforms are mostly going to be defined by their exons that they have. That's what the identity of the isoforms usually come. Because once you get an isoform, if you think these are functional, they have to get translated to proteins. Again, because we are working with that particular domain, there are something called long non-coding RNAs and non-coding RNAs, where that part has some, everything is non-coding in some sense, right? So that length actually sort of starts to kick in. But for all other sense, it's actually just the coding part of that particular gene. Yes. Which part? This? Oh, that's because there is an alternate starting exon. So, okay, that's a good question. I did not actually give you a preview of that, right? So, this is my entire DNA, but the transcription can actually begin here, here, or here, anywhere, right? This is also determined by where the polymerase starts. So there's something called TSS, which is transcription start site. What is the transcription start site? I think it's a very good question, which I should have highlighted earlier. Right? So this is one, right? 2, 3, right? It could be any random site. Now, what is happening physically at this step, right? So at the physical step, when this is your DNA, right? At this point, I don't know what is what, and the polymerase also doesn't really know. What it will see is it will come here and it will start scanning things. I will just make this as a polymerase. It will see if there is enough sort of proteins here lying in its vicinity. So for initiating the transcription, it requires what are called IFs, initiation factors. So if it sees, as it is scanning, and if it sees enough synergy here where the binding can happen along with the initiation factors, it can start synthesizing from here. And that's your one possible site. In some cases, depending on where the things are sitting and what is sitting in its vicinity, and again, all of this is in 3D, it could happen that at this point it doesn't have enough initiation factors for it to start beginning the transcription. It could also happen that you are, let's say I take my liver and again I take my kidney, and I look at the same DNA sequences, right? So the DNA sequences are the same. This is ATG, actually let me not make it ATG, but AAC. And again, in my kidney also it's AAC. So it comes here, but it doesn't have in this is my liver and this is my kidney. It doesn't have enough context for it to actually start transcribing yet. So it will do it slightly later here. And my isoforms will start to form from here. So this is where the transcription is beginning, and that's what determines the length that you have of the untranslated part, right? So it still hasn't started making protein yet, because you haven't actually even figured out the RNA. This is where you're beginning to initiate the end of the beginning end of your transcription. So that's what the 1, 2, 3 is different. Thank you for asking that question. I did not give enough context. Okay, this can happen across cell types also, that in certain cell types, same DNA, I begin my transcription here, or I could begin it here, I could begin it here, right? So the way to figure out is there are assays to actually tell you where the transcription initiation site is. It's called, forgetting the name, 1 second. It's one of the things which always comes out from one institute, and I'm totally blanking out. But if you search for Riken TIS, which is Riken is an institute in Japan. What's it called? I think it's called Race Seek or something. I'm forgetting, but maybe I'll come back to it in the next class. Those essays actually tell you where is the initiation site of your transcription. Yeah. There's a question. Hmm. Hmm. Batch to batch. By batch to batch, you mean that today. So mostly in one cell type. So that's a very interesting question because the answer to it is like I don't know, because it depends on the context. A lot of it is determined based on the context, right? So certain diseases can happen because of you getting more of this isoform versus this isoform. It could happen because the way the joining happened was not correct. There are like multiple possible ways things can go wrong. But in a normal human being, usually the isoform structure is more or less expected to be constant. So whatever is my isoform level, usually, right? Like usually, again, all with this caveat that it doesn't change so much with respect to time. Of course, you'll have one slightly higher appendance of one isoform versus compared to other. Also, you don't get pure isoforms, right? You will have a bunch of both these kind of isoforms in your body overall. But usually that ratio, usually again, will remain constant. But there are diseases in which it becomes differential, including, of course, cancer. It will become on the spliceosome machinery, right? Those spliceosome machinery, which we'll talk about. Okay. Still thinking about that name. So since it's troubling me, I will Riken transcription. Cage, not race. So it's called Cage, cap analysis. No, now I remember. Cap analysis of gene expression. So it essentially tells you at which form, where does the initiation begin. Okay. Okay. So a single gene can give rise to many mRNAs. This is actually an example taken from your body. One single gene, this gene can either be called a calcitonin or a CGRP. It has exon 1, exon 2, exon 3, exon 4, exon 5, exon 6. When it is in your thyroid, right, exon 1, 2, and 3 and 4 are retained, and it makes what's called calcitonin. Whereas the same gene, right, the same gene is being determined based on these boundaries. It, in your nervous system, is called CGRP because it retains 1, 2, 3, 5, and 6. So see here, not only is the N codons different, the first three are retained, but the N codon choice is different. It retains 4 versus it retains 5 and 6, and that changes the entire protein that you're going to synthesize. This is one example of a thing which all of us have inside our body. Okay. How did we figure out where did they came for the same thing, right? So one way to figure out is, of course, they share the same structure of exons that you have. So once you do transcriptomics, you will try to map it. Where are these sequences coming from, right? And in this case, exon 4, when you map your DNA, when you do DNA sequencing, well, you do not do DNA sequencing, but you do RNA sequencing, this will get mapped here, whereas this one will get mapped to 5 and 6. So we know that nowhere else is there this tight structure which is shared. Yes. So that is your DNA sequence. Your human genome that you downloaded, right? HG38, GRCH38, which is your 38th version of reference sequence that we have, that has both exons and introns because that is the full DNA sequence. Yes. Oh, good question. Can we know whether what was the immature version of this sequencing? Yes. How will you get it? Yes. Right. So I haven't even talked about the technology yet, but yes, you can. Um, right now, if you so most of the technologies when I talk about the technology, right? So this is your cell wall and this is your nucleus. So whenever you any technology that you talk about which tries to capture RNA, you're working with the cytoplasm part of the cell. So what I will do in my experimental setup is I will add a detergent, and you literally add detergent and the cell walls will break. Your RNA will come out. Now this is cytoplasmic RNA where the splicing has already happened. But if I want to be super creative, what I will do is I will add one more round of extreme level of detergents, and I'm going to burst the nucleus out, and I will get what's called the nuclear RNA, which is, in your words, the immature transcripts. And there I will have the introns partly retained. So I can actually do this analysis. Okay, but this is slightly way ahead because I have not talked about the technology. No, you cannot. Yes, but I don't know what part of my DNA will become RNA. I don't know that. Hmm. So you can do a targeted version. Is that what you're saying? Hmm. You can, but slightly harder to do it, right? Huh? There will be a lot of combinations, and you'll have to figure out where to begin and where to end. Yeah, so it's much harder experiment to do, right? So if each single base of our DNA gave rise to RNA, yes, you could do that, but it won't happen. So you cannot do that, right? So you have to figure out which part of the DNA will give you rise to RNA, right? So if the problem is a lot of this DNA actually is just sitting there, like nothing is going to come out from this DNA, like nothing, right? So broadly, that number stands at whatever that number is, 98% or 96% or 92%, depending on the context. But there's nothing happening. It's not resulting in mRNA, and of course, it's not going to result in proteins. So why is it sitting there? And in a famous paper or an infamous paper in 2012 or even before that, it was broadly termed as the junk DNA, right? This is junk because it's not really doing a function. However, we know that's not correct because it's not just sitting there. It's actually helping you figure out what should bind where, and can you help express things? Can you, again, this is not linear, but the way this works is if a protein comes and sits in this region, which is broadly called transcription factors, this is written as TF, it will bend and it will interact with this site where the initiation could begin, and it actually helps proteins start the transcription. So it's not just sitting there, but it's actually helping you. We, for a long time, we did not know where are those regions, but we now know where the initiation happens and where does it end and so on, right? So simply you cannot sequence DNA to figure out where are the introns. Make sense? Okay. Oh, sorry. Yeah. So this is another example, NTNG1. It has 10 different exons. Exons 5 and 1, 2, 3, 4 are constant. 5 to 9 can be spliced in or out. And that's why it gives results. It results into these isoforms, right? So what are these isoforms, right? So isoforms are nothing but the joined exons broadly. In this case, for example, 8, 9, 10 are joined. This has 8 and 10 joined and so on, right? There are multiple possible combinations. One single gene, this becomes combinatorial in some sense, then it can give rise to different isoforms. If it is giving rise to different isoforms, it also, by definition, is also giving rise to different proteins. Okay? So from one single gene, you can synthesize different proteins overall. Why are we not able to splice out different exons? Yeah. So for splicing certain things out, you require a very special signal, which is called the splicing signal. We don't actually discuss this too much in the class, but I will give you a very quick perspective. So this is exon 1 and exon 2. If I have to splice them out, or if I have to sort of join them, then one of them needs to be a donor and one of them needs to be an acceptor. So this is a splicing donor, for example. This has to be a splicing acceptor. This happens because there is a special signal, and I'm forgetting what that signal is, but I will just make it up on the go. that this has to have certain AA sequences repeated, and this has to have certain UU sequences repeated, and the spliceosome machinery should be able to recognize it. If it doesn't exist, you cannot splice it out. Basically, broadly, that's why certain things can remain as it is. Okay? So it's kind of actually written in, that's what this particular slice shows. They have a GU rich actually. That's the signal that I was looking for. In this case, this intron, the splice site has a GU rich signal, and this one is AG rich, not AA and UU, but very close. And that allows you to see which ones will join to each other. And that's how they recognize also that which are exons. And I think this was the question that you asked, which is the exon and which is the intron, right? So um, most introns will have this kind of structure overall, which is AU, AA, or AT, AC, and so on. That's kind of repeated overall structure. But again, this is biology, so there will always be exceptions, but broadly, this is what it recognizes overall. So it's a pattern recognition machinery which is able to recognize what to splice out and what to keep, right? So most introns will have this as their first base and this as their last base, and that becomes whether you can actually remove them or not, or you can join them or not overall. This is what I was talking about. What is the mechanism of splicing? So for splicing, you require a donor site and an acceptor site, right? So we'll not go too much into the biochemistry, but very briefly, what the splicing machinery is trying to sort of do is making this, basically breaking this end. So this becomes your donor, this becomes your acceptor, this is going to roll over, and the moment it rolls over, you can also do it's just like making another H2O bond and releasing it. This gets released, it will come here and join. That's what the splicing machinery broadly tries to do. Of course, this is a simplified version. It's not as simple as what is depicted here. The reason this becomes possible is because of what's called the spliceosome machinery, which is basically another set of enzymes or proteins broadly, which will enable this kind of reaction to happen, right? So they are able to sort of see that there is a GU, I should do an attack here, break it. Once I break it, the entire molecule will roll over. I will have more stability if I go and join these two so that I get this particular version of the isoform, right? So there's a lot of magic in some sense happening. By the way, we don't still understand a lot of how splicing machinery works, what is their recognition pattern, and how does it differ across cell types. A lot of it is actually unknown. This SNRNPs, which you had seen in the first slide, this is where this particular portion of it is responsible for doing the function. So they will bring the two proteins together, which is which enables this spliceosome machinery to work. But hopefully, broadly, this idea is clear how the attack is happening and how the exons are overall joined. Any questions about this? Oh, where do they go? I guess most of it will undergo decay. Um, so at least for exons, I know there is something. The process called nonsense mediated decay (NMD). If you have only some portion of your transcript synthesized and it has to be decayed, that's what it will come in. Which particular machinery acts here, that I don't know, but all these will be degraded and recycled. So you can reuse your nucleotides. You don't go waste. How exactly that degradation happened, that I don't know, but these are degraded and recycled. You can detect if there are very small molecules just lying around in your. There's a surveillance mechanism which always is surveilling for small molecules, so that will detect it and start degrading. Oh, yes, it is. It is how much. So this spliceosome machinery is also kind of the abundance, the relative abundance of their spliceosome machinery. Like there are certain genes which will, of course, which make the spliceosome machinery. So the relative abundance of of them will tell you how much proteins, which particular isoforms, like, let me formulate my thoughts 1 second. So depending on their relative abundance of the spliceosome machinery will tell you which isoforms will be more abundant or less abundant based on how efficiently they can carry out this reaction. Because I only told you about these two sites, but it's a slightly larger site that most of the spliceosome machinery recognize. Now that recognition happens because you have those proteins which can recognize it. So if in a cell type I don't have that many which I can recognize from this end, I will probably keep these two exons. But if in certain cell types, if I have more of them, I'm going to splice them out. So it's a cell type specific thing or a tissue specific thing. Okay. Okay, so it should be precise, but of course, right, you can have errors in the process, and broadly there are two types of errors that happen when the spliceosome machinery is trying to do its job. You can end up actually skipping an exon. For example, in this case, you should have retained exon 2, but you end up skipping it. It happens in a lot of diseases. This can result, this is broadly called exon skipping, and it is something which happens in a lot of diseases where I have a disease just because I don't have exon 2 present in my RNA. You could also have a cryptic slide structure that the machinery, rather than attacking this end, this being the acceptor, this being the donor, it recognizes, because it's a pattern recognition machinery. So if that GU structure happens to be within the exons, it will recognize it, it will create the attack here and not here, and it will lead to this cryptic site selection. This is actually very common in certain cancers. These are called fusion transcripts, where you fuse two different exons and make a totally different protein in some sense, right? Whenever this happens, you're making a new protein overall, and that does happen in at least a few cancers, if not all of them. Okay, so hopefully that was clear as to what is why is it complicated, and why we should try to measure RNA, and when we do, what kind of things can go wrong, what are we trying to essentially do, right? How would you quantify? Hopefully this is also clear that while we have 2 copies of DNA, only a portion of them will get transcribed to RNA. So let's say this is gene 1. It can have 3 copies of mRNA. I can have gene 2 can have 6 copies, and gene 3 can have again maybe 4 copies. The amount of protein is directly proportional to the amount of RNA that you synthesize. From 2 copies of DNA, for gene 1, I can make 3 copies of isoform, right? These, by the way, could be the same isoform or could be individual, could be different. We will come to that problem in a bit. But for now, just assume that gene 1 has 3 copies of mRNA, gene 2 has 6, and gene 3 has 4. Given whatever you learned about DNA sequencing, how will you capture this? How will you capture the molecules, the RNA molecules? So I told you that if I wanted to do DNA sequencing, I will take the DNA, shred it apart, and throw this off on a sequencer, and I will get my sequencing part, right? All of you understand how a sequencer works now, so you know how sequencing happens. If I were to do the same thing for RNA, what should I do? Can I use the same technology to do it? Yes, no. If no, why not? If yes, why yes? No. Okay, why not? Hmm. 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 Okay, so let's chop it off. Okay, so let me chop it. This one off. This one off. This one off. Huh. So. Yes. So this gene 4, so whenever, yeah, no, so that's a good question. Let me clarify, right? So let me draw the structure and it will become clear. So let's keep it very simple. This only has 2 exons, and this is a totally different gene, which means it has totally different exons. Okay, they don't share the same exons. So let's say this one has E1 prime and E2 prime and E3 prime. So this full stuff is DNA, right? If I join everything, this is joined all anyway. This is full stuff is DNA, but there is a soft boundary here that this is making one kind of gene, this is making another kind of gene. Okay, so I should call it gene 2, and it has 4 copies or 5 copies. This one has 4. I want to quantify this. I want to measure. What is it that I want to measure? I want to measure if how many copies of mRNA does gene 1 have, how many copies of gene 2 does this have. How will I, can I use sequencing to do this? No, protein you measure in proteomics class. You cannot measure protein in this class. You cannot measure. Not there yet. Not there. Maybe next year when I teach this, I will talk about it. Not there yet. Proteomics, you cannot do this in this class. Unfortunately. Not because of any other reasons, just because it's not there yet. How would you measure this? You have access to a black box, and that black box, you have that black box is your Illumina, for example. What does Illumina do? That if I input a sequence, which is DNA, it can spit out the sequence of this. Using that black box technology, I want to measure the sequence, but I also wanted you to tell me the abundance of. Is the problem clear? In DNA sequencing, we had not talked about abundance because we only had DNA. It's fine. I just want to know the sequence because I know there are only 2 copies mostly, and that's the problem that we cared about. I only taught you half the thing because while there are 2 copies, again, because diseases are so complicated, some diseases happen because of 2 copies in certain chromosomes. You have 2.5 copies, or you have 4 copies. Diseases happen for that reason also, but I did not teach you that. In most cases, we cared about mutations, so we didn't care about the abundance. Here, I want to know the sequences. I want to know where they came from so that I can tell you that this came from gene 1 and gene 2. And I want to also measure how many of them are there. How would I do it? Can we group them? No, you can do it in proteomics. You can do it. Proteomics, that's what happens. You know what you have to measure, you go and you measure. This is totally unknown. That's why it's hard, very hard. Proteomics, it's very, what you have to measure, go and measure. Why CDNA? Nobody knows here what is CDNA. Do you want to explain the CDNA? Why should I make DNA? RNA is good enough, right? Okay, so that's step 1, right? So what is the problem with this technology? So I did not even actually motivate the problem, right? The reason this is hard is not just because this is, this is a dual fold problem, but the reason it's hard is because the moment you do anything to these are the most unstable species that exist, one of the most unstable species that exist in your cell. The moment you do anything with them, they will start to break apart, right? So that's why we can sequence the DNA of like dinosaurs and like, previous, very extremely old people, our ancestors and so on, which are buried deep under mummies and all. You can do DNA sequencing, but very hard, also almost impossible to do RNA sequencing because it would have decayed by now. Very, very easy to decay. You give it like, throw it on this, on this wall with slightly heated, it's gone. Full molecule degraded. So we can solve that by using Aniket's solution, which is to make cDNA copies. What is cDNA? Is nothing but just you have an RT enzyme, a reverse transcriptase enzyme, which will come here and convert this RNA to cDNA. It's not doing anything interesting. It's literally making a copy. But when it is making a copy, that U that you have in your RNA will become, broadly it will become T. And for all other practical reasons, which we don't have to get into, this will be more stable version of the same copy, right? So We have solved one level of the copy. But how will I identify this? Like how? But then we are again going to back to proteomics. No primers. Full throughput, high throughput, as high throughput as you need. How will you clone it? Primers, no primers. Full throughut. I can take any organism on this Powai campus and still be able to do it. That's the power of this. So let me give you some I'm trying to jump a bit too much, but let me give you some more facts about what makes this particular um, uh, species. Yes, go ahead. Yes. Okay. Fair enough. So the first thing that you do is you have to be able to pull down these molecules, right? Don't forget that I'm teaching a class in RNA, but there is a lot bunch going on in the cell. There are so many other molecules that are floating around, including what? Including the functional non-coding RNAs, which is 96%. But the reason these mRNAs, I'm right now only interested in, practically mostly I'm interested in things which are hopefully going to code for proteins, and they have a very special signal. They have what's called a poly-A tail. So when I said that the DNA gets converted to RNA, there is one more step actually that happens, and by the way, it's not very simple. Like this arrow itself is like 5 or 10 different steps that have to happen. There has to be capping, there has to be restructuring, and so on, which again for this class does not matter. But there's one more step that happens is at the end of when it is done copying the mRNA, it will keep going and it will make all of them have a poly-A tail towards the end. That helps you actually identify that these molecules are your mRNA and not the other remaining 96% or 98% or whatever that number is. I think it's 96%. So that you only capture things which are going to code for proteins and not the ones which are otherwise functional. There again, the 96%, they're also very important, but the hope is that any disease or anything that we're interested in is likely coming from that 40%. So the first thing they have is poly-A tail. And that's one of the things that the technology that I'm going to just talk about right now, which is broadly called RNA-seq, tries to resolve, right? So no primers required. All you require is poly-As, which are there in all the eukaryotic. It doesn't work in prokaryotes as easily, but in all the eukaryotic RNA molecules will have a poly-A tail attached to it. So we are not attaching anything to the coding part yet. We are broadly doing what's called a pull down. So there is you can, like we get slightly into the biochemistry of it, but you can do a particular reaction which will allow you to literally separate the ones which have a poly-A as compared to all other molecules, right? So that reaction, that pull down is possible. So how does this poly-A? So you are doing a reverse complement, for example. Like I'm not talking about that specific process that we do, but you can think about that. If you had a sequence which had poly-T, you will come and it will start sort of hybridizing here. You do a pull down from there. So you can literally, if this is your molecule floating around, right? You can just add primers, right? Like primers, but they're not the interesting primers. They are like just T repeated. Once you detect this, you will do a pull down. A pull down means that just separate those out. Because poly-As are the only ones which are present at the end of your mRNA. All mRNAs have this shared property that they all end with poly-A tails. That's why. Okay. Okay. So that's what this technology, which is called bulk RNA sequencing, tries to do. it first will recognize that there are molecules which have this poly A tail. You will do a pull down. How does that pull down happen for this class? It doesn't matter. There's a ubiquitination step and so on. Again, none of this matters for this class. But you can separate this out. And once you separate them out, you convert them to cDNA, right? This is what we did here. So first step, we figured out which molecules we want to separate, convert them to cDNA. Once we convert them to cDNA, we will repeat our step, right? Which is, we'll fragment it. So we will again cut it apart. And we'll throw this off on a sequencer, because at the sequencer level, we don't require any innovation, because it knows how to handle DNA. This is DNA at this point. So you have taken the RNA, you have figured out the mRNA molecules by figuring out the poly A tails, you have converted them to cDNA, and you're literally sequencing it. And this data that you come out from here, from there, we go to tell you that this came from gene 1 and this came from gene 2. And today, for the rest of the class, we will see how to do this problem computationally. The problem is clear? Any questions? Why is this problem hard? That's the most interesting part, right? So the reason this is going to be very, very hard is, we already talked about this, right? So this is your DNA. This is your pre-mRNA, which has the introns, which will get spliced out, and this will form your mRNA. This is what is happening in vivo, which means that those 3 steps happen within the cell. Now, in RNA-seq, what you're doing is you're doing it outside the cell, so you're doing it in vitro. The first step that you're doing is you're taking those RNA fragments. You are doing an RT, which is reverse transcription. You are making these double-stranded DNA molecules or cDNA fragments, and then you're sequencing it. This is something that's happening in vitro, which is you're doing everything that has happened inside the cell. I rupture the cell, all these molecules come out, I sequence them by converting them to cDNA, and I get a lot of sequences data. Now comes the interesting part about what this class is about, which is to figure out how to process this data so that I know where does this sequence came from and what is its relative abundance. How many copies of each sequence is doing what. In the process, I also want to figure out, are there new isoforms that my data has, because I'm going fully in a high throughput and in an unsupervised manner. I want to figure out which isoform is more abundant, which isoform is less abundant, and so on, all from this data. Okay? Yes. This one, this is mostly for illustrative purpose. The fragmentation actually does happen after converting to cDNA. This is only just showing you the exon level stuff, but the fragmentation does happen afterwards. Do you think we'll lose the information about splicing? Are we losing information about splicing here? Good question, actually. The other way to ask this question is, can we recover how it was spliced out? Right? Yes, right. So can we recover? And that is where what the and hopefully I'll get to it, because that is the crux of what the class is. Yes. From a molecular biology problem, we have again switched it to a computationally or a computer science or a statistics problem, right? We have done everything. We have done the molecular biology part of it. All that is great. We got sequences. Now it becomes a computation problem, and that is for us to solve. So this is a typical computational workflow of how to process RNA sequencing data. And all these blue things that you're seeing are name of different tools, that blue, green, and yellow. We will not cover all of them because some of them have become defunct. We don't have to cover them. But broadly, what I want to show you is there are a lot of phases in which the data has to be processed. First, you align it, or you can assemble it, you quantify it, and finally you normalize it and you model it. This class, hopefully by the end of this 25 minutes that I have left, we will cover about how to align this data. And then maybe on Thursday we can do a hands-on. I know that you have a lot of exams, so I don't want to pressurize you by talking about stuff on the last day of the class. So we will do a quick hands-on of how to handle RNA sequencing data. And It will be again a reverse Bollywood approach in the sense that we will not talk about how we do this and do this. Well, this today we'll talk about, but this we will not talk about. Once you come back after mid-sems, we'll talk about what are the methods there. Okay, so what is the problem, right? What is the problem that we have, right? We have seen one version of this where there are two mapping problems. You can either assemble it. So if I had a lot of reads, I can try to assemble it and try to figure out where it came from. Or I can try to use a reference to figure out that this reads, where did they came from? So this is a mapping problem. This is an assembling problem. In assembling problem, I don't have access to the reference. I'm creating it as you go along, right? So it's like those diagrams or those videos, if you have seen, if you're as old as me, is none of you are, is rail, there's an engine driver who's putting the rail as he goes along, builds a track. This assembly problem is very much similar to that. Where the reference problem is you already know your way and you're trying to figure out where these things came from. This we already did in DNA sequencing, and we will again not focus on the assembly problem. We will focus on the mapping problem. Okay, now is a question time. So you have seen this data, right? You have seen, you want to figure out where it came from, which gene it came from, and how many copies does it have. Can I use BWA or any algorithm to do this? Or do you think we require to innovate here? Do you think the DNA mapping algorithm that we did use in the previous class, can we use it, or will we require some innovation to be done here? So I will just erase this just to give you context of what is it that we want to do very, very quickly. And I will keep this diagram. So what data we got? We have like a lot of these are all reads. At the end of the day, what does matter to me? I want to, for example, if I'm calling this as gene 1 and if I'm calling this as gene 2, I want to know how many number of reads aligned to gene 1, whatever that number is, how many of them aligned to gene Y. Ideally, I would also want to know, in this case, let's say this only has one isoform, which is E1 and E2. In this case, I also want to know how many isoforms do you see of this type and how many isoforms do you see of E1 and E2. Right. And of course, in this G1, there is only one isoform, so it doesn't matter. So I also want the quantification of how many of these, how many of these. Question is clear? How would you solve this? Okay, what would the local alignment do? Yeah. Good analogy. Okay, very good. Okay, any other thoughts? So here, what is the problem, right? Like, I cannot take this read. Well, I can take this read, of course, and try to align it as if it was my DNA sequencing data. But I'm also interested in this problem, which is I want to figure out that if I got these reads, how many of them are for this isoform, how many of them are for this isoform. And in this process, what am I discounting? So I'm discounting that what Ananya said, which is in E1, there is no E2, and in this case, there is no E3. So in your mapping, when I'm mapping, if I want to map these reads to this, I will have to take into account that this does not exist. In this case, I will have to take into account that E2 does not exist. So that has to be done by the algorithm. Is this a simpler problem or a harder problem? Aligning RNA sequencing data, is it simpler or is it harder? Would you require any more information that I can help you with? If somebody gave you RNA sequencing read, what other information will you ask for, which will help you to figure out the right mapping? The cDNA, which is for all, okay. So this by cDNA, you mean the Okay. So let's say this is your entire DNA. Okay. I give you this. Will that be sufficient? Or do you need more? Any other information you would need? Nothing else. No, but you can ask. Okay, so you will require some gene map, right? That I gave you DNA, which you already have, all of you have seen it. All of you have done that exercise where we downloaded one full chromosome 11. I did not tell you, by the way, there that until we did the towards the end, where is the HBB gene, the hemoglobin beta globin gene? Where is it located? I need coordinates. I need to figure out that this is the starting of gene 1 and this is the end location. Nothing else, but I need to know where are the genes actually located. Why? Because I don't want to waste my resource trying to figure out what is the right mapping for this to other things. If somebody has already figured out that gene 1 sits here and gene 2 sits here, I might as well use that information, right? So that's correct. So we need that information. Now with those two information, let's try to see what can we do. So aligning, when we did aligning DNA sequencing data using BWT, right? What is it that we tried to do is we created a Burrows-Wheeler transform of your DNA sequencing data. You took each individual read and you figured out where does it begin from. The problem, if we want to do this with our RNA data, is it's very hard. In this, everything was continuous, whereas in RNA, I need to be splice aware. I need to be aware that splicing can happen. Exon A can join to exon B, but exon A can also join to exon B. Plus, there are gaps in between, which are introns. So this aligner, whichever this aligner is going to have a very uphill journey. It's a very, very hard problem because it needs to be, once I get this read, it could have come because these two exons have joined. In your DNA, they are very far apart because they have an intron in between, but they could have joined just because this read, because this particular transcript has exon B and exon C, right? Is that problem clear? So any alignment that you want to do should be aware that this kind of splicing can happen. I could have read coming from, half of the read could map to this exon, half of this could map to this exon, just because when I actually processed it, they were joined. So if I do my fragmentation, I could get some reads from here. And then half of it will align here, half of it will align there. Okay, very, very hard problem to solve, right? Very, very hard, extremely challenging. What is the problem? So given read, I would like to quantify how many copies of each isoform are present and how can I assign my isoforms, how can I assign each read to an isoform unambiguously, right? For example, to go back, actually I have one figure which will make it clear. Yes. So what is it that makes it clear, right? So imagine this is your, these are your individual reads. If it came from only one location, it's okay. You can go and map there. But what I also want access to is, I want to tell you which particular isoform among, this is your gene, this is your transcript X, which is your isoform X, isoform Y. I want you to be able to also tell me that whether this read, this small read, did it come from this isoform or did it come from this isoform, right? So here, for example, if you see something which is in towards this very extreme, I can, in this case, I can tell you that this came from the red isoform. However, if you have reads which are kind of, if anything originated from here, right, I would know because in my reference, I know I have access to these isoforms. These are annotated. In the sense, somebody has actually gone and done this annotation on the human genome that tells you that for this particular gene, these are the only two isoforms that we see in any individual person. So the only possibility for this particular gene to happen is either in the form of X or in the form of y. But when I get the data, I want you to tell me how many of x's are there, how many of y's are there. Why is it complicated? Because these two share some exons. If my read originated and I mapped it here, I would know definitely it came from the second isoform. But if I get a read from here, it could have either come from x or it would have come from y. Okay, so that is the complicated part. So how is the mapping done? So in the case of, we'll quickly cover this, and then I will motivate a problem to think about. And very quickly, I will not ask questions whatever I teach on Thursday, but I will motivate you to think about that problem before you leave. It's a puzzle, right? So it's a good note to end on, which I did not intend to, but I think I went much slower, so we will end earlier. But overall, what does star, it's an alignment tool, tries to do is, it says that let me use suffix arrays. Let me use the same idea of suffix arrays, but be very, very smart about it. So think about, these are your exons which exist in your genome. You can create a suffix arrays using only the exons, right? You all know what are suffix arrays. Where does it begin from? Where does it end? So you have access to sequences. So what is a suffix array? Is nothing but this is my suffix, and let's say this is my location. So I know, for example, if I see a sequence of ATG in my read, it came from 13th location in my DNA. What it does is it will take the read. This is your read. It will take part of the read and try to see in the suffix array, does it match to anything? It will again split the read and will try to find out that, can I find the occurrence of that particular string in my suffix array of the exons? And if you are able to locate part of it, then you have found something. You will rotate. You will now proceed to the next half and see. Of course, it could have come from the same exon, but it's also possible that it could have come from any other exon. So when you run this star, and at some point when you do the project, you will have to run this algorithm. This is broadly what it is trying to do. It has figured out that, okay, part of this read comes from this exon. Does the other part that I have in my read, can it be explained by any other sequences that I find in my exons? So we have seen some iterations of this when we were doing DNA sequencing as well. Because in DNA sequencing, what we talked about was, if you want to map without mismatches, if you want to map with mismatches, then you will take part of your reads and you'll see whether that can align. You will allow for a mismatch, and then you will continue iterating over this idea. Star does very, very simple. Star handles that in a very, very similar vein, which is it looks for reads. It looks for part of the read, where did it come from, and tries to do this iteratively. And in this process, it has figured out the location for each of those reads. Why are we doing this? Why is it complicated? Because this read that you got, it will not map here. This is intron. It will map to part of this, and it will map to part of this. And that's what it's trying to figure out overall. Okay? This is what star does. There's another tool which is called it's called Salmon. The idea behind Salmon is also very, very interesting. It says that you can take all the isoforms that you have, and you can create. The reason I'm covering this on the surface is because these are just intuitions that I want you to get, and then the third solution is what we will start to go into the details. So here in Salmon, what you do is you take all the transcripts, you separate them by a dollar operator. This is your isoform 1. So you know the actual sequence. Again, you know the actual sequence, and so on. On this particular long sequence, you run this idea again of suffix arrays. You store a hash table. Everyone knows what is a hash table. It's a simple lookup table, basically, right? So just like your suffix array, it's a very simple lookup table. You take the read now. You have created a suffix array of this transcriptome. So you know where it begins, where it ends, and you know where are portions of the suffixes coming from. You take the read and you figure out which of these portion does it align to. right? So for the way it works is, rather than working with base to base, it has a fixed k-mer size. A k-mer is a sequence of length k. In most cases, this is 31. So you take your read, you take the first 31 bases, and you go to your suffix array and see where is it coming from. It has its particular location. So for example, you took this part of the read, which is your k-mer. You figured out that this comes from this starting location and this overall ending location. Now you can go there and then start your mapping. So the difference between this and star is star will do it individually, where it's storing the suffix array of your individual exons, whereas in this case you are storing a much compact suffix array of your transcripts or your isoforms, which are separated by which are separated by your dollar operator. So it is in essence much faster to run, because the suffix array size is going to be much smaller, because you have around I'm forgetting the number, but let me recall. You have around 100,000 isoforms. So you're only storing 100,000 entries in your table, whereas the number of exons are of the order of, you can multiply that by a order of 4 or 5. So in the previous case, I think 3 exons per, forgetting the average number, but anyway, I'll correct this number next time. But 400k exons, for example, the lookup is at least 5x faster if you do quasi mapping. This is called quasi mapping, because you're mapping base to base, but you're starting by first having figured out that which isoform should I even try to map to, rather than in star where you have to figure out which of these individual exons your read came from. So it's slightly smarter as compared to star overall. Yes. Yeah, so the hash map, right, which is basically, so when you have this k-mer, this is coming from your read. Now, when you store the suffix array, you're storing suffixes also of the length equal to 31. So that 31 length will give you a map of, given this k-mer, which is the suffix, where did it come from? Which isoform did it come from? So you have the address of that particular isoform. Given a particular read, I'll run a quick lookup. It tells me that this is the isoform that you came from. Now go there and start mapping, rather than having to do it at the individual exon level. So that's what makes it much faster. No, these hash tables are for k-mers. So each what is the lookup, right? So the lookup is again very similar to this. So if I fix my size to be k equal to 4, okay. Now k equal to 4 is a g a c a g g g and so on. It stores that likely it came from transcript 1 and transcript 2. This came from transcript 2 and transcript 3, and so on. Yes, all the genes, yes, right? So the input to this is your DNA sequence, is what's called the cDNA sequence, which is all possible isoforms that have been seen in humans in your genome overall, right? So one gene can have 5 isoforms, it can have 10 isoforms, or it can also have 1 isoform. So you take all those sequences, concatenate all them together, separated by a dollar operator, and then you create your suffix array from that sequence. Okay, we will come to this solution 3, but before that, I will, since we have, since we are short on time and will not finish, let's talk about a coin toss experiment, and you will see why we're doing this in the next class. So imagine I gave you, I want to figure out, you're doing a simple coin flipping experiment where you're given 2 coins, A and B. You don't know whether they are fair or not, so they can have any probability, theta A and theta B, which is what I call it here, of landing up a heads. What I want is, what is the probability theta B and what is the probability theta A? Okay, 2 coins, let's say they are biased. Their probability of landing heads is theta A and theta B respectively. I want you to help me calculate or guess what theta A, or design an experiment which at the end of the experiment will tell me theta A is equal to whatever 0.51, theta B is equal to 0.49. Very abstract experiment, right? Very weird also, like why who would do this? How would you go about it? And you will see why two coins in just a bit. So so if you want to simplify it, think of it as maybe for now, if I gave you only one coin, what experiment will you do? Keep tossing it. Okay, so fine, keep tossing it and yeah, measure the outcome, right? Yeah, so I have a coin, very simple, right? I have a coin. I will toss it 10 times, 5 times it lands heads, maybe 0.5 is what is theta A. I can do it again and again and again and again, right? So I can of course figure out what is the theta A probability with it, right? So in this case, it's you could do the same thing with coin B, right? Because I told you what is coin A and what is coin B. In this case, simply that you did it for one coin toss. You can do the same thing for two coin tosses because I told you that before giving the coin that this is A and this is B. You can repeat the same experiment and calculate what is theta A and theta B. And this intuitive guess, which all of you did right now, is what's called maximum likelihood estimator. Very complicated statistical name, a beautiful sort of entry point for doing statistics, which we will not do in this class. It's called MLE, estimation. Whatever you guessed intuitively in this case works. It tells you that this is the right probability for A and this is the right probability for B. Now, I give you again two coins. I remove the identity and I ask you the same question. So at each experiment that you're doing, right, I'm giving you two coins. You can do whatever you want. I'll take it back again from you and I'll give it back to you again. I want you to tell me what is theta A and what is theta B. Or A and B does not matter because I've not given you the labels, but I want you to tell me what are the two probabilities. What experiment will you do? Is the question clear? Yes. So I again have two coins. Okay. I want you to tell me what is the probability of them getting heads, each of them. But when I give it to you, I don't reveal the identity that this is A, or they look very similar, or they look exactly similar, or they look exactly the same, or they look the same. Not to blow it off, right? So they look the same, or identical is the right word. Yes, they look identical. And I still want you to tell me what is the probability of them. So you cannot exchange them, or you cannot sort of figure out what is the identity associated with it. What are the two probability of them getting heads? What are their individual probability of landing up with the heads? How would you think about this? Yeah. Is the question clear? Slightly abstract, but so think about the isoforms. Your reads came from those isoforms, right? So when I drew these two isoforms, like you don't know, right, which isoform came from where, and you got one read, and I want you to be able to assign it to one of them. How will you do it? Same thing. You are able to see that I'm tossing two coins and I'm getting heads and maybe tails, or heads and tails, or whatever. But I don't know the identity of each of them. Is it A or is it B or what? Is it isoform A, isoform B, given a particular read? Which one should I assign it to and why? Any ideas? Does the question make sense? Everyone? Somebody who doesn't, this question doesn't make sense? Two identical looking things, two identical looking uniforms, two identical looking coins. I toss them and I see something, and I want you to tell me which one is A and which one is B. Something is different definitely about those two coins that I'm telling you offhand, that there is some distributional property differences between them. Their theta A and theta B are different. That I can tell you. Okay, that I can guarantee you they are not identical. But when you look at them, they look very, very similar. You have to do an experiment to tell me how will you guess which one has, let's say, higher probability of landing with an heads. Yes. But no, but yes, fair enough. What you're saying is fair enough. He's saying Aman, right? So Aman is saying that you can do an experiment where I gave you the data, you will collect the data, and then you'll do machine learning on it, right? Which is you will collect the data, and tomorrow you'll come and predict. And I hate machine learning. I just don't understand machine learning basically, right? So how will you do it then? It's not that true that I'm not don't hate machine learning, but in this context, I think it's not applicable. Yes, you know each of these isoforms. Yes, you know each of these isoforms, right? But they are for other reasons. How will you assign whether it came from this one or it came from this one? This is theoretical possibility. This is A, this is B. Okay, so let me show you. Actually, I don't want to reveal this. One second. Hmm. Sorry, say that again. Let me show you. Actually, yeah, good question. Let me skip over and show you. Okay, this is what it looks like, right? So don't figure out. Don't worry about the E step, M step. Don't worry about that. But this is what it looks like, right? So um, there are three isoforms here: blue, green, and red. This is your genome. Okay, these are the different reads that you get: A, B, C, D, and E. I want you to tell me, did A come from blue? Did it come from green? Did it come from red? Which of these isoforms should I assign? I have to count the number of isoforms, right? I have to count for each isoform how many reads are there. Should I assign A to blue, green, or red? Should I assign B to blue or green? Because it can overlap with these two. C is simple because, well, no, C is not simple because it can be blue or red. D kind of is simple. Why? Because it has a split thing, and that split thing is only in this particular isoform. D confidently I can assign to the third isoform. What about E? E also could be from green or red. Okay. How would, yeah, okay. But I don't know to begin with. I don't know which one is more. They're all, if I don't know anything, I don't know isoform A, B, I don't know isoform blue, green, and red, which of them is more common? Uh, A is itself a read. This is a read. I'm asking you that when I do the counting, that this isoform has 5 copies, this isoform has 2 copies, this isoform has 3 copies. Should I count A as part of blue, green, or red? Yes. Right? So tomorrow, when I want to quantify the abundances, what is my naive hypothesis? Null hypothesis is all of them are equally abundant. Now, this is my naive hypothesis. But when I actually do the measurement, I want to be able to refine it, right? This is what I started with, but is it true, right? I don't think it's true, right? Because you can see that D only came from red. So it has slightly more evidence only towards red. So it's not that I can, or in other words, like if I have to assign A to one of these isoforms, which one should I assign it to? Join the reads. No, you cannot do that. Reads are small, right? So this read is fixed, length is fixed. I want you to tell me whether this read would have come from blue, green, or red. And you don't have to do a hard assignment. You can also tell me that there's a probability of 1 by 3 that it came from blue, green, or red. But at the end, what I want is I want a matrix of uh, red, blue, green, number of copies that this has. This has 5, this is 3, this is 2. What is it, that number? Or 5.5, 3.5. What is that number, right? So your total number of reads are, let's say, 10. So among the 10 reads, what is the relative abundance of isoform blue, isoform red, and isoform green? And I know I have exceeded time, but just a thought experiment. I'll not upload these part of the slides because I want you to think. Why is it similar to that coin toss experiment, right? So imagine I had two coins. I do an experiment. I take those two. I do 10 coin tosses overall, and in one of my tries, I get 5 heads and 5 tails. This is my trial number 1. In my second trial, I take, so I have 2 coins, right? I don't know their individual identity and what separates them, but I can choose one of them. And the second trial, again, I don't know whether I chose this one or this one. That one I don't know. But I get 4 heads, 6 tails. Does it tell you anything about your original source, which you're not able to observe, but you can observe what the output is? You cannot observe the identity of this, but you can still observe the outcome of this experiment, that choice, and the answer lies there. Okay, so I think it's a good stopping point. It's the same problem, by the way, just to it's the same problem that, is this A or is this B? Is this red, is this blue, or is this green? Same problem. Very, very similar problem. Okay, so I'll end it here. I think the portion for mid-sem will not cover, will cover whatever we talked about today, but will not cover things which we're going to talk about on Thursday. Okay, so you're not under the pressure of doing anything on Thursday. Thursday, I do want to cover the lecture part of it. So I will cover the first half, and then we'll do a hands-on analyzing RNA sequencing data set. Okay? But do think about this problem, that how will you solve this? It's a puzzle.